Hi everyone, my name is Jayoung Kim from Cedars-Sinai Medical Center and New City. Today, we will share the paper from our lab entitled Proteomic Profiling of Bladder Cancer for Precision Medicine in the Clinical Setting, a review for busy urologists. Okay, at present, proteomic methods have successfully identified potential biomarkers of urological malignancies, reflecting different number of key cellular processes. In this paper, we will summarize the current emerging technologies for advanced discovery, targeted proteomics, and proteomic application in bladder cancer, particularly in discovery of human-based biomarkers.
Recent genome messenger RNA expression analysis demonstrated that BC can be classified into different molecular subtypes. These different molecular subtypes of BC have distinct progression patterns, biological and clinical properties, and response to chemotherapies. There are currently 5 published classification methods. As you know, that these include guidelines from the University of North Carolina, which is UNC, MD Anderson Cancer Center, MDA, and the Cancer Genome Atlas, TCGA, London University, London, and the Broad Institute of Massachusetts Institute of Technology, and Harvard University, Broad. Proteins are effector molecules that mediate the function of genes, and their DNA regulation contributes to the pathogenesis and therapeutic resistance of many diseases, such as cancer and neurodegenerative disorders. They represent enormously valuable resource for personalized diagnosis, prevention, monitoring, and treatment. Therefore, the protein properties such as abundance, post-translational modification, stability, localization, transportation, and interaction with other molecules all have been intensively studied. Histologically, the proteins were previously studied in individual basis. The proteome is very complex and information-rich research. The human genome contains about 20,000 protein coding genes. In comparison, human proteome is much more complex and may contain over 6 million proteoforms. The major research for proteoforms diversity include first the single nucleotide polymorphisms and mutations at the DNA level. Second, alternative splicing and RNA editing at RNA level. And third, errors in translation and post-translational modification, which is called PTM at the protein level. Among these, the biggest contributor of proteoform diversity is phosphotranslational modification, PTM. Currently, over 200 PTMs, such as phosphorylation, ubiquitination, glycosylation, have been characterized according to the UniProt database. Do you know the difference of the discovery versus targeted proteomics? Okay, the proteomics can be broadly classified into discovery and targeted proteomics, which are highly complementary to each other. The discovery proteomics is generally used in hypothesis-free and comprehensive profiling studies to identify novel protein complexes as well as differentially expressed protein and modified or localized protein. It is a very powerful tool for identifying novel candidate biomarkers and therapeutic targets and for providing fresh biological insight. In comparison, targeted proteomics is generally used to quantify the candidate proteins of interest in a much larger cohort of samples with a higher quantification accuracy and precision. So, can you apply the proteomics to study or understand the bladder cancer in our cases? Yes, we are doing it now. In clinical setting, the urinary protein profiling can be used to support diagnosis, monitoring, and stratification of personalized diseases, while tissue proteomics can address the personalized therapeutic intervention by identifying new therapeutic targets. Up to now, several urinary biomarkers have been described as including FDA-approved immunoassay, which detect the urinary level of BC-associated antigen, which is called BTA-STAT, and the nuclear matrix protein 22 and MP22 with modest performance. In case of NMP22, 68% of sensitivity and 79% of specificity, and BTA-STAT, 61% sensitivity and 78% specificity. Using two-dimensional electrophoresis 2DE, people found that PGAM1 was significantly upregulated in BC compared to adjacent tissue. In a parallel approach, cystatin B was also found to be a prognostic biomarker at the tissue level. Increased expression level of proteins were correlated with the stage grade and recurrence and progression. Compared to urine analysis, there are relatively few studies on blood-based BC proteomics. People examined the serum of patients with low-grade and high-grade BC and healthy controls. They found that there was differences in the expression of 5 proteins, among which S100 calcium-binding protein A8 and S100A9 could distinguish low-grade versus high-grade BC. The authors further confirmed the finding in a later study by analyzing serum sample from 108 patients with BC and 52 healthy controls. They found the expression of S100A8, S100A9, S100A4, and CA1 decreased in BC, and the expression of annexin 5 increased significantly after surgery compared to patient before. Like serum, saliva is a complex mixture of proteins with concentration in excess of 10 order of magnitude. The progress of getting saliva is the least invasive and simplest. The saliva proteome is consistently changing from birth to adulthood, so age must be considered in the reference data. At present, the study of saliva proteomics is mainly focused on oral disease such as oral cancer. There are also several reports about the relationship between salivary proteomics and gastric, breast, and lung cancers. However, there are no reports on saliva proteomics in BC. Therefore, the potential proteomic BC markers in saliva samples remain to be confirmed. The proteomics markers identified in different human samples were summarized in this table. So in this paper, we discussed about several things. First, the proteomics is expected to improve personalized medical treatment by better assessing disease risk, more accurately monitoring disease, and improving targeted treatment. Second, the addition of proteomics profiling-based biosignatures will provide tremendous benefit to the existing molecular phenotyping of bladder cancer patients in deciding treatment protocol, monitoring responses to the therapies, and recurrence of a screening of a high-risk individual. For successful clinical application, definitely prospective multi-center-based randomized trial should be considered. Utility of proteomic phenotyping combined with genomic phenotyping will play an increasingly important role in research and clinical management in conjunction with standard of care. Thank you.